The aim of this video is to give a brief illustration of how a research hypothesis can be tested by proceeding through the different study designs, and in doing so to highlight some of the parameters that characterize the different types of study designs. So a hypothesis or theory can often be tested using a range of different types of study design, and when this is the case, you sometimes find that the evidence from studies on humans for a particular hypothesis develops chronologically, starting with studies near the bottom of the hierarchy of evidence and finishing with experimental trials. And this happens in part because of cost, moving from less expensive hypothesis generating studies at the bottom to more expensive and robust conclusive studies at the top. And as the evidence accumulates, there's more justification for testing it with a more robust and sophisticated study design. So here's an old example concerning maternal fish oil consumption and birth weight. And the initial hypothesis for this example was generated by an observation made by the author when he was a junior doctor, that mothers who had delivered high birth weight babies seem to have a preference for whale blubber in their diet. So it's a bit like a case series. So in order to test this hypothesis, a first step might be to do an ecological study using existing and routine data sources. And by doing that, it makes the study cheap and quick. Now, an ecological study uses aggregated data from different populations rather than individual level data. So in this case, the unit of observation might be a country, for instance. And we may be able to obtain national average information on birth weight from each country to use as the outcome, and national level data on the cells of fish, or perhaps national surveys about dietary habits that could be used as the exposure. And in such a study, we might expect to see higher birth weights in populations with higher fish consumption. However, the evidence would be weak because there may be a range of other differences between populations that are also linked to birth weight and that are causing the correlation at this population or ecological level. For example, there may be differences in genetics between populations, or smoking levels, or gestational weight gain, etc., etc. If that were the case, then the relationship seen at the group or ecological level would not be seen at an individual level, and this is called ecological bias and is something we have to be aware of with ecological studies, and it's what makes it a potentially very weak source of evidence. Of course, we wouldn't know if the result was biased or suffered from this ecological bias, but we'd need to collect individual level data to find out. So we might then decide to do a case control study, collecting information on individuals. Here we might take a random sample of high birth weight cases and a random sample of normal weight babies from the same source population that generated the cases, or the high birth weight babies, and then try to describe the mother's dietary habits during pregnancy. We might also collect information on potential confounders to try to control for this in the analysis, or we may even be able to match the controls to the cases on variables like age and sex of the baby. In doing so, we're trying to control for confounding, but doing this in the design of the study rather than the analysis. And we might then be able to verify that there is a relationship between fish consumption and birth weight at the individual level, but we'd probably have concerns that there is unmeasured confounding. And we'd also be concerned about how the controls were selected. For example, is there a selection bias in the sense of sampling controls from really healthy sections of the population? Or perhaps there might even be some recall bias in the information that we collected about dietary habits from the woman who gave birth to the high birth weight babies. That's causing a bias in the results. So in summary then, we may have added to the evidence with this relatively quick and cheap study, but we would still have concerns about the quality of the evidence in support of our hypothesis. So a next step might be to do a prospective cohort study where we identify a target population, that could be all pregnant women, perhaps recruiting a random sample at the point of the first antenatal clinic, So we're taking a random sample before the outcomes occurred on a population at risk. And we could then monitor the diets of these women prospectively over pregnancy at defined intervals and follow up the babies at birth to measure their birth weight. And in analysing these data, we could perhaps split the women into groups defined as either low, medium and high fish intake, and then compare the birth weights in these groups. To make the exposure groups comparable, we would measure potential confounders and try to adjust for them in the analysis. So in this study design, we'd have better information on dietary habits that's not affected by recall bias, but we would still be concerned about residual confounding by other factors that influence fish intake and high birth weight. So finally then, we might consider doing a randomised controlled trial as a robust test of a causal effect of fish oil. The source or target population might be all pregnant women again, and we'd have to think about what the intervention should look like. It would be difficult to randomise a whole food-based diet, but we could randomise a dietary supplement, perhaps marine fatty acids. The study could be blinded, and we would be pretty confident that our randomisation would ensure comparability, so there'd be no confounding. And this sort of study would provide the highest level of evidence in terms of internal validity. But in our example, we'd also be concerned about the compliance in the two groups with respect to drawing conclusions, and we'd also question the external validity. For example, we've only looked at a single dietary component, and we may have only managed to recruit normal weight, health conscious women. And lastly, we might also wonder whether we've missed the etiological window with respect to when in pregnancy the supplement was given, or even before pregnancy. So you can see from this example how a hypothesis can be investigated using a sequence of designs ranging from cheap and quick to more expensive and more logistically complex. However, the payback for more complex designs is the potential for greater internal validity and accuracy, or the potential for removing biases that will affect our ability to draw conclusions about a hypothesis. So to finish off, in discussing this example, we've highlighted several design parameters that characterise the different study designs. So first of all, the unit of observation, whether that's individuals or populations. And secondly, how the exposure is allocated to individuals or groups. Is it random by experiment, or is it done by nature when we just observe? And the timing of observations. Since exposures and outcomes often occur over time, we typically want to capture exposure and disease over time, like in a cohort study. But sometimes this is not possible or is too expensive to do. Now, a related aspect to that is when we should try to measure the exposure. What is the etiological time window? If we're interested in reproductive and developmental health, then this schematic shows how exposures and outcomes may occur and act intergenerationally right across all different windows of life across the life course. So it's very important to think about how and when exposures and outcomes are measured and define that when designing a study. And we also need to think about the source population. What is the population that we are sampling from? And we often have to specify eligibility and exclusion criteria to define this source population. And then how do we sample from the source population? Do we sample before the outcome has occurred, as in a prospective cohort study? Or do we sample from known cases and none cases after the outcome has occurred, as in a case control study? And there's other options as well. We could just take some form of random sample at a particular point in time or snapshot in time without knowledge of whether the outcome has occurred or not, and that's a cross-sectional study. Lastly, do we need to collect information on other covariables to control for confounding? So these are all aspects to consider in designing and interpreting a study.